Lloyd-Smith. Great. Hello, everybody. And I'll try to make that go away. There we go. I'm Jamie Lloyd-Smith. I'm in the Department of Ecology and Evolutionary Biology and also Biomath. And I'm going to be telling you about our work on infectious disease surveillance. This is actually a project, it's a couple of years old and published, but it's the closest thing that we have in my group to a software tool. Most of what we do is mathematical and computational modeling on a sort of problem-specific basis, rather than developing tools intended for others to use. Anyway, this was work done by a very talented postdoc named Chris Strelayev, who's now moved on. He's doing work at UC Davis now. And I'll frame the project for you, or the problem for you, as follows. So most of the work in my group pertains to the ecology and evolution of infectious disease, and in particular, zoonotic pathogens. So zoonoses are infections that come out of animals and infect humans, and this is where we get most of our new and scary pathogens. You see here we've got swine flu, HIV, SARS, MERS, Zika, the list goes on every year. And it's also where the great majority of current human infections come from. We share a lot of pathogens with animals. There's a lot of public health incentive to understand the ecology of what these pathogens are doing on the animal side to help us understand the health risks. So here's a little cartoon. There's some pathogens circulate around in animals, they spill into humans, and then they may get going in humans as well. One very important task is to try to track disease levels in the so-called animal reservoirs as a component of how we assess the risk from these diseases. So that's important, but it's also very challenging because there are a huge number of pathogens out there, a huge number of different animal species we could be looking at, and then the data that we can get from any given species tend to be messy and incomplete. So it's often not a big data problem so much as a small to middle-sized data problem, but we want to make the best possible use of those data. So the general problem is that in many disease surveillance settings, we only have limited access to the host individuals we're trying to sample. So you can get them only during certain seasons or certain life stages. You can sample, say, piglets, but not adult pigs or some kind of wildlife species at a time when they're in an accessible geographic region. We typically can only sample cross-sectionally, so we only get one sample from each individual, which is much less informative than longitudinal sampling. And then the sampling intensity is often erratic or opportunistic or, I would say, spasmodic. If something interesting happens, everyone jumps on it and samples. But we tend not to have long-term consistent time series. And so gaps in the data are a problem that we have to wrestle with frequently. The goals in what I'm going to show you are to maximize the information gained from whatever samples we have been able to collect and analyze. And in general, the question is to understand as much as we can about how the diseases are transmitted, what we call the transmission dynamics, and see if we can make inferences from the data we have about the periods or regions that have not actually been sampled directly. We'd often like to fill in those gaps. And the idea in what I'm going to show you is to look at multiple types of data that are collected from individuals when they're sampled. You can take a blood sample and a urine sample or a saliva sample, get a bunch of kinds of data, and then exploit the biological connections between the data that you can get from these different sample types, and then perform a joint statistical analysis to pull the most information possible from the data that you've obtained. So that's a general problem. In particular, I'm going to be talking about two types of disease surveillance data. And these are virologic data. So you're actually looking for the virus or its genetic material in samples you've taken from animals. And this might be by culture or by PCR. And this gives you evidence of current infection of any given individual who's been sampled. And on the other hand, there's serologic data. So here you're looking for specific antibodies against a given pathogen using an ELISA, or there are all sorts of other assays that we use. And this gives you evidence of past infection with that pathogen. So altogether, these two assays give complementary information, and they're linked by biological mechanisms in terms of the course of infection and immune response within an individual. But typically in the field, they're analyzed and reported independently. So they're not integrated and analyzed in an integrated fashion. The case study I want to tell you about pertains to a long-term surveillance data set for swine influenza in China. Now, you guys may recall when we had the 2009 pandemic of influenza, it came to us from swine, probably in Mexico, But that revealed to us there's actually a tremendous lack of information about what influenza viruses are doing in pigs. This despite the fact that pigs have long been recognized to be a really dangerous, quote-unquote, mixing vessel for influenza viruses and the source of several past pandemic strains. So there's a big gap in what we know about how influenza transmits among swine. But this is a really unique data set which extends well before the 2009 pandemic, goes back to 1998, and it is samples taken from pigs as they pass through a slaughterhouse, an abattoir, in Hong Kong, but the swine were actually mostly grown and farmed out in various provinces of China and shipped into Hong Kong. So it gives us a bit of a window into what's going on in Chinese pigs, which we're not able to access directly because the Chinese government has not, to that point, been cooperative with sampling. So here's some raw data. This was reported in a Nature paper in 2011. These bars, if you can see them, mark the frequency of virus isolation. The different colors are different strains of influenza, and this is over the span of this 11-year period. And you can see there's actually, you go from a period of higher rates of isolation to a lower period later in the decade. And so it looks as though there's less influenza circulating as you move forward through time. But the study also collected serological data. So this is antibody evidence that individual pigs had met flu before they were sampled in the slaughterhouse. And if you can see down here, you'll just have to take my word for it, the seroprevalence, which is what we call the fraction of pigs who are carrying anti-influenza antibodies in their blood, the seroprevalence did not decline over that month. So you got the virus isolation going down, the seroprevalence is not obviously going down. So that's where that study left it. I learned of this and met the people involved, and we got involved to see if we could learn more about the epidemiology of swine influenza in Chinese pigs from these data. So in particular, there's the opportunity to try and fill in the gap. We've not been able to collect data from the actual pigs on the farms in China, but we can try to infer what's going on by thinking about the nature of the data that have been collected. So there are some pros and cons to the data set. First of all, very rarely, very unusually, this is a long-term systematic surveillance effort in a livestock species. These are all too rare, and it's a great gap in our knowledge of what pathogens are circulating, and even in livestock species where we do have regular access to them. It's also an 11-year time span, which is a lovely long period of time compared to usually there'll be a sort of a focal response around a particular outbreak that might last a year, and then people get bored and move on. So those are the pros. On the negative side, these data were actually collected for this virologic strain surveillance, so they weren't sampled in the appropriate numbers and structures to support the kind of analysis we're trying to do. There are big gaps in the serology data. Serology antibodies are actually only measured for 3 individual years within this decade, and in between we don't have any direct information. And then, of course, there's no information from within China. We're just left with what we see in these pigs when they arrive in Hong Kong. So the approach we took is to take advantage of the biological coupling between the two types of data we've got. And so you can look at experimental infection studies where they give influenza to pigs and see how the infection proceeds, and you can map out the course of the response and how it's measured by these two assays that we've used. So there's a period of about 5 to 7 days where you tend to get positive hits via PCR. You can find evidence of the virus in the pig's nose. So that lasts about a week. And then just around the time that that's going down, not coincidentally, antibodies are coming up. And so if you take a blood sample more than a week after a pig's been infected, you can get a positive hit using these serologic tests. So we've got these two time courses which describe how the biology of infections unfold. And it just happens that those time frames basically match the recent history of these pigs before they got sampled. So the last 5 to 7 days they spent in transport, so they're trucked across China, which is a big country, and that tends to take anywhere from 2 to 5 days. And then they spend time in what is colloquially called the pig hotel. So they're sort of gathered in a large holding facility before they're moved by train into Hong Kong for slaughter. So that matches this period of time that they would be PCR positive for the virus. And then their whole lifetime before then, they spent on a production farm. So whatever their farm of origin was is where they spent all their life up until they got shipped off to slaughter. So we constructed a Bayesian state space model to try and put these pieces together. And this is a hierarchical model. I'll walk you through this picture in some detail. So it's a state space model, which means we're separately thinking about the actual process, that's the biological process that's underpinning the structure of the data we're collecting, and separately from that, the observation process itself. So we call the observation process the data model, and that consists of just binomial or multinomial sampling from whatever the true population distribution of disease is. And the process model, which I'll go into some more detail on, describes the actual transitions in the disease states in these pigs. And then there's a parameter model underpinning all of it, which is basically a set of priors and ultimately posteriors on individual parameters. So the process model, it's got a couple of layers. And I'll just focus here. There's one layer that links up down here the probability that pigs are getting exposed either on the farm or during transport during any particular window of time. So that's the actual epidemiology is happening in this part of the model. And then this up here is the probability that a given pig that you sample is actually going to be seropositive or virus positive at the point that you sample them. So that's one layer, and then there's a temporal element where you have a change in the actual process, the actual exposure probabilities from month to month, which reflects how the disease may be intensifying or weakening through time. So that's the broad model, and I should say we just model that using a simple Markov chain assumption with very little structure. We just essentially let it drift and let the data tell us what happened. So we put all of this together into a big MCMC framework, and just very crudely, this takes in the time series data for both the virus isolation and seropositivity data, prior distributions on all the parameters and initial conditions, and it spits out posteriors for all of our model parameters, including the missing data, including these periods of time where we weren't actually able to sample the pigs. So here are the results. We've got virus isolation data at the top, antibody seropositivity at the bottom. I'll just pull out the main stories. What you've got here is the dots show the actual raw data fractions. This is the fraction of pigs who were sampled who were positive for influenza virus through time. And then the line in shaded region shows the posterior, which was inferred by the model. And here you see a comparison between the raw frequency and the posterior frequency. And you see the posterior is actually a slightly smooth version of the raw data. It's sort of all the fluctuations are ironed out a bit by integrating with the serological data. And that leads to a bit less variation than the raw data exhibit. Down at the bottom, you can see the time course of inferred seropositivity, so what fraction of the pigs had antibodies reflecting previous exposure to the virus. And here you can see there are periods where it's very well defined when we do have data, and in between, we actually do impute the missing data, but the confidence intervals are quite a lot bigger, or credible intervals, I should say. And again, there's slightly less variation in the imputed posterior data than in the raw data, but they do agree with an uncertainty. So what do we learn from this in sort of big picture terms? By filling in these gaps and imputing the missing seroprevalence data, we can actually identify that there is a significant rise in seroprevalence over this 11-year period. So rather than the disease declining, as the straight virologic data seem to show, we're actually seeing more pigs have been exposed to influenza by the time they get to the slaughterhouse. We can actually use this and the duration of time that pigs spend in each compartment to calculate the hazard rate of infection, so effectively the intensity of transmission, both during the time spent on the farm and during transport and holding prior to slaughter. And we see here, this is the farm estimate here and the transport and holding. The first thing we see is the hazard is actually higher on the farm than during transport. And second, we see that it's been increasing on the farm over the decade, whereas it's been holding steady during transport. So just to sum up what I've told you, we developed this Bayesian state space model to jointly analyze these two streams of disease surveillance data. We applied it to this unique 11-year data set of flu surveillance in pigs in Hong Kong. The main findings were that the decline in virus isolation rates did not reflect a reduction in disease, as was initially thought, but actually it was because more pigs are getting infected earlier in their life when they're still on the farm, so they're actually immune by the time they're in transport, and so they can't be carrying the virus when they get slaughtered. We detected a clear rise in flu exposure in pigs on Chinese farms over the span of this 11 or 12 year period, and showed that the risk of infection is higher on the farms than during transport. And this actually runs contrary to expectations in the agricultural field, where they talk about transport stress giving rise to more infections. But in this case, it seems like the pigs are getting infected quite happily on the farm before all that. And finally, with an eye to this being a software tool, this method is very generalizable, so it can be applied to any parallel streams of surveillance data where we have some knowledge of the time course of how the different assays evolve over the course of infection. All right, with that, I would like to thank, again, Krista Laff was the postdoc who did the bulk of this work, a few very valuable collaborators who gave us access to the data and funding sources, and you for your attention.